Welcome to the deep dive. So today we are getting into something that honestly completely changed how I picture the immune system. We're exploring its hidden architecture and looking at how aggressive cancers actively demolish it.
Yeah, it is a truly fascinating shift in perspective.
Right, because usually we're just, hyper-focused on the cancer cells themselves.
Exactly. But our entire discussion today is grounded in this really detailed 2026 paper from Nature Cancer. And the researchers used, cutting-edge single-cell and spatial mapping to look deep inside human lymph nodes.
Which is so cool. And our mission for you listening today is to uncover the microscopic, city planning of our lymph nodes. We want to figure out how certain lymphomas hack this system to tear down the walls.
And understand why realizing this architectural collapse is happening. Why it could completely change how we think about cancer progression.
And treatment, too, because usually when we picture our bodies fighting off a disease, we immediately jump to a battlefield metaphor.
Right, it's all brute force.
Yeah, exactly. Like immune cells are the brave soldiers, cancer cells are the invaders, and they're just clashing in this chaotic, muddy, microscopic war zone.
We tend to focus almost entirely on the combatants, like who is fighting whom and who has the bigger weapons.
But looking through the lens of this new spatial biology from the paper, that battlefield metaphor feels, entirely wrong. We are not looking at a muddy field at all. We are looking at a bustling, highly engineered metropolis.
We really are. And to really wrap our heads around how this metropolis collapses under the weight of disease, we first need to walk through how a healthy immune city is built.
Right. So for you listening, think of your lymph node not as a random squishy bag of immune cells filtering fluid, but as a perfectly zoned metropolis.
A very strict metropolis.
Extremely strict. It has distinct districts. You have a commercial district where the B cells hang out, multiply, and do their specific jobs, and then a residential district reserved strictly for the T cells.
And that spatial organization, that zoning, is the absolute foundation of our immunity. If the B cells and T cells aren't in their specific correct locations, the lymph node simply cannot function.
It just fails.
It completely fails. It cannot mount an adaptive immune response.
Yeah.
But what is so striking here is that the cells defining the structure, the ones drawing the maps and enforcing the zones, they aren't the immune cells themselves.
Oh, wait, really?
Yet they are a tiny fraction of non-hematopoietic stromal cells.
Okay, let me pause on non-hematopoietic because that is quite a mouthful.
It's a bit technical, yeah.
Meaning these cells aren't born from the blood stem cells in our bone marrow, right? Like our white blood cells are. They're structural cells, the actual city planners.
They are the architects of the lymph node. And the paper focuses on a few key types of these planners. First, you have fibroblastic reticular cells, or FRCs.
FRCs, okay.
They form a sort of three-dimensional mesh network, almost like the steel girders of a skyscraper. It acts as a scaffold that also sponges up fluid.
Like the structural frame of the city.
Exactly. And within that network, you find a specialized subset called follicular dendritic cells, or FDCs. And those are located specifically in the B cell zones.
Okay, got it.
And then weaving through all of this, you have the endothelial cells that make up the blood and lymphatic vessels.
Which are basically the actual highways moving traffic into and out of the city, right?
Right.
So if they are the planners, how do they force the immune cells into these perfect districts? Because obviously they aren't building physical brick walls that the microscopic B cells are bumping into.
No brick walls. They build chemical walls. They use a class of proteins called homeostatic chemokines.
Okay, let's break that word down. Chemo meaning chemical, kinesis meaning movement.
That is the perfect breakdown. You can think of these homeostatic chemokines as the chemical street signs and GPS signals of the lymph node. They constantly broadcast location data.
So, okay, if I am a B cell and I have just traveled down an endothelial highway into this lymph node city, how does that GPS system actually guide me to my specific district?
Well, those follicular dendritic cells we mentioned, the FDCs, they sit in their specific district and pump out a very specific chemokine called CXCL13.
CXCL13, okay.
And this creates a concentration gradient.
Yeah.
It's highly concentrated near the FDCs and gets weaker the further away you get.
Like a radio signal getting weaker as you drive away from the tower.
Exactly like that. And your B cell has a receptor on its surface called CXCR5, which acts essentially as a highly tuned GPS antenna, specifically looking for that CXCL13.
Oh, I see. So my B cell enters the lymph node, my little CXCR5 antenna picks up that CXCL13 signal, and I don't know, drive straight toward the strongest part of that signal.
You follow the signal right to the source.
And that chemical gravity pulls me and millions of other B cells tightly into these specific neighborhoods, which are called follicles.
You got it. And while that is happening, other stromal cells over in the T cell zone are pumping out entirely different chemokines, specifically CCL19 and CCL21.
Okay, so different signals for different cells.
Exactly. The T cells have antennas tuned to those signals, which pulls them into their designated residential areas. It creates this exquisitely balanced, perfectly guided flow of traffic.
Okay, let me make sure I'm fully wrapping my head around the baseline here before we introduce the disease. Our healthy lymph nodes are these incredibly well-zoned cities.
Right.
The structural stromal cells are the architects, and they use chemical GPS signals, these homeostatic chemokines, to guide the immune cell traffic into the proper neighborhoods so they can do their jobs.
Exactly. It is an invisible web of chemical gravity holding everything together.
Which is just wild to think about. But I guess that brings us to the core investigation of the paper. Like, what happens when cancer moves into this neighborhood?
Right, because it really disrupts everything. The researchers contrasted two very different types of nodal B-cell lymphomas just to see how this architecture holds up under pressure.
Let's look at the patient experience for a moment to ground this. The first cancer they looked at is follicular lymphoma, or FL.
Which is clinically an indolent disease.
Right, it's slow growing. Like a patient might just notice a painless swelling in a lymph node, and doctors often take a watch and wait approach because it progresses so slowly, sometimes over years.
Yeah, watch and wait is very common for FL.
So when we look inside that FL patient's lymph node, what is happening to the city?
Well, the spatial organization mostly survives. The malignant B cells are multiplying, so those B cell follicles, the commercial districts, they expand and get much, much larger.
They swell up.
They do, but the fundamental zoning laws remain completely intact. The street signs are still broadcasting, and the distinct separation between the B cell zones and the T cell zones is preserved.
So the city is bloated, like the population has exploded and the commercial district is completely taking over the map, but it's still undeniably a functioning city with clear borders.
Exactly. It's crowded, but orderly.
But that contrasts sharply with the second disease they analyzed, which is diffuse large B-cell lymphoma, or DLBCL. This is the highly aggressive form.
Very aggressive.
For the patient, this usually presents as a rapidly growing mass, often accompanied by severe night sweats, fever, weight loss. It is fast, and it is destructive.
And when the researchers looked at the DLBCL lymph nodes, they observed a catastrophic loss of tissue organization. I mean, the clear boundaries are completely wiped out.
Just gone.
Just gone. The distinct follicles vanish. What is left is a chaotic, diffuse mass of proliferating malignant B cells, T cells, and macrophages, all just blended together without any structure.
Wow. But, if the city boundaries are entirely destroyed in DLBCL, my immediate thought is that it must be passive destruction.
Like they're just getting crushed.
Yeah, exactly. Like the malignant cells are just multiplying so incredibly fast and aggressively that they act like a bulldozer. They just physically crush the city limits by sheer physical force and crowding.
And honestly, that is the assumption many would make. But when we connect this to the deeper spatial and genetic data, we find something far more calculated.
Oh, really?
Yeah, it is not a bulldozer tearing down the walls. It is an inside job targeting the city's communication network itself.
Wait, if it's an inside job, that implies the city planners, those structural stromal cells, are somehow compromised.
They are.
But how could you possibly look close enough to see if a structural cell has been hacked? I mean, you are looking at a microscopic piece of tissue with literally millions of cells.
Well, the researchers utilized single-cell RNA sequencing.
Okay, let's break down single-cell RNA sequencing for you listening, because usually if you want to see the genetics of a tumor, you take a biopsy, grind it all up, and sequence it, right?
Bulk sequencing.
It's like throwing a fruit salad into a blender and trying to figure out what was in it based on the taste of the smoothie. You know there's apple and banana in there somewhere, but you don't know which specific piece of fruit contributed what.
That is a great way to put it. Single-cell RNA sequencing allows you to look at every single piece of fruit individually.
Which is amazing.
It isolates each individual cell and reads the exact RNA transcripts it contains. RNA is the messenger. It tells us exactly which genetic instructions that specific cell is actively reading and executing at that exact moment.
So you're not just looking at what the cell is, you are reading its active to-do list.
Exactly. And when they read the to-do lists of the stromal cells, our city planners in the DLBCL lymph nodes, they found profound physical changes.
Look, what kind of changes?
These cells transformed into what the paper classifies as remodeled FRCs and remodeled endothelial cells. Their genetic instructions had fundamentally changed.
The hackers got to the architects.
They absolutely did. And the most critical change was that these hijacked planners simply stopped producing the homeostatic chemokines.
Oh, wow.
Yeah, the CXCL13, the CCL19, the GPS signals that keep the city organized, they vanished from the to-do list. The structural cells were no longer broadcasting the map.
Okay, let me play devil's advocate for a second here. We are assuming the signal stopped. But could it be that the cancer cells themselves are the ones that are broken?
How do you mean?
Like, maybe the malignant B cells just mutated and lost their CXCR5 GPS antennas? So they are driving blind, ignoring perfectly good street signs.
It is a good thought, but the researchers tested that exact hypothesis.
Yeah.
The malignant B cells are not broken in that way. They proved that the DLBCL B cells still retain their CXCR5 receptors right on their surface.
Oh, wait, really?
Really, they even took these cancer cells out of the patient and put them into a lab setting to observe their behavior using a Transwell assay.
Okay, how do you test a cell's sense of direction in a lab dish?
So picture a tiny lab dish with two chambers separated by a microscopic porous filter.
Okay, picturing it.
They put the malignant B cells in the top chamber and a high concentration of the CXCL13 chemokine in the bottom chamber.
And what happened?
The malignant B cells migrated right through the filter toward the signal. They can still follow the directions perfectly.
So the cars are functioning perfectly. The hackers didn't cut the brake lines or blind the drivers. They hacked the city's actual GPS system and took down the street signs.
That's exactly it.
The B cells are fully capable of following the map, but the map no longer exists.
And actually, the situation is worse than the map just disappearing. Ectopic sources of chemokines appear.
What does that mean, ectopic?
It means coming from the wrong place. The researchers found that a specific subset of exhausted T cells within the tumor microenvironment suddenly starts producing CXCL13 instead of the stromal cells.
Okay, so not only are the official street signs gone, but random pedestrians are standing in the middle of the street holding up fake detour signs, pointing traffic in completely random directions.
Yeah, that creates total navigational chaos. The signals are scattered. They're coming from the wrong places at the wrong times.
Okay, for everyone listening, let's solidify this mechanism before we look at the cause. The complete collapse of the lymph node in aggressive lymphoma isn't from physical crowding or an expanding tumor acting like a bulldozer.
Right.
The cancer creates a microenvironment where the structural cells are actively reprogrammed to turn off the zoning signals, and at the same time, random cells start throwing up fake signals.
Yeah.
So the immune cells get completely lost, and the physical structure of the city just dissolves.
Which brings us to the most complex question of all.
Yeah, the why.
Right. If the structural cells are being hacked and reprogrammed, what is actually hacking them? How does a malignant B cell reach out and change the genetic to-do list of a fibroblastic reticular cell?
I mean, the cancer cells must be secreting some kind of malicious virus or chemical payload to brainwash the city planners, right?
Actually, it doesn't do it directly. The hackers, well, they are actually the body's own defense system.
Wait, what? The immune system is hacking itself.
Sort of, yeah. When the tumor starts growing, tumor-reactive T cells enter the lymph node. Their job is to recognize the threat and eliminate it.
Right, standard immune response.
But when they see the massive DLBCL tumor, they realize they are facing a severe threat. They start secreting a powerful distress signal called interferon gamma, or IFN gamma.
Okay, IFN gamma. That is a classic, potent inflammatory signal. It's basically the biological equivalent of pulling the fire alarm.
It is a massive fire alarm. And this IFN gamma triggers a chronic inflammatory feedback loop.
Oh, I see where this is going.
The stromal cells, our city planners, they hear this blaring alarm. And their programming dictates that during a fire, you don't worry about normal zoning laws. You react to the emergency.
So in response to the IFN gamma, they shut down their normal day-to-day city planning chemokines. They stop worrying about keeping the B cells and T cells in their neat little neighborhoods because the city is metaphorically burning down.
Exactly. Instead, the stromal cells start pumping out emergency inflammatory chemokines, specifically CXCL9, CXCL10, and CXCL11.
Okay, emergency signals.
Right, and the sole purpose of these emergency signals is to rapidly recruit as many immune cells as possible to the site of the fire.
But, I mean, I am struggling to see why this is a bad thing. If the T cells are pulling the fire alarm and the city planners are calling for backup to fight the cancer, shouldn't that help clear the disease?
You would think so.
Why does this help the cancer survive and tear the node apart?
We have to look at the difference between acute and chronic inflammation. In a normal acute infection, say you get a nasty bacterial infection from a cut, inflammation is incredibly helpful.
Right, it puffs up, gets warm.
Exactly. It is acute, meaning short-term. It temporarily pauses normal tissue function, floods the area with immune cells, kills the bacteria, and then the alarm turns off.
The inflammation subsides, the planners go back to work, and the city rebuilds. The fire department puts out the fire and goes home.
But cancer isn't a short-term bacterial infection. It is a persistent growing mass. It provides a never-ending alarm.
Oh.
The T cells keep screaming, the IFN gamma keeps flowing, and the fire alarm never, ever turns off. The lymph node becomes permanently trapped in an inflammatory, dysfunctional state.
So the ecosystem is torn apart by the very sirens meant to save it.
Exactly. It's a tragic irony.
The aggressive lymphoma survives by trapping the immune system in a perpetual state of emergency panic. Because the immune cells are in a constant, uncoordinated frenzy, they can never organize a methodical, effective attack against the tumor.
They're just running around in chaos.
The villain doesn't need to fight the police. The villain just ties up the dispatch lines with constant fake emergencies so no one can mount a coordinated response.
That is exactly what happens. And the researchers didn't just stop at observing this tragic irony. They needed to definitively prove that this invisible chemical rewiring was truly the root cause of the physical collapse seen under the microscope in DLBCL patients.
Well, how do you prove that causality? I mean, you can't just freeze time inside a patient's lymph node and ask the cells why they are moving erratically.
You can't. So they turned to advanced mathematics.
Math. Okay.
They created an in silico model, which is a highly sophisticated computer simulation. But the brilliant part of their methodology is that they fed the computer only the chemical data. They gave the algorithm what they called chemokine attraction potentials.
Okay, let's unpack chemokine attraction potentials. What exactly is the computer looking at?
Well, imagine a massive spreadsheet. It simply lists which cells were producing which chemokines and which cells possess the receptors to hear them.
Just a list of senders and receivers?
Exactly. The algorithm received absolutely zero pictures, no spatial data whatsoever. It had no information on where the cells actually lived or originated in the physical tissue, just the raw chemical text messages being sent and received.
Wait, by reading just the chemical text messages, like who is texting whom and how loudly, you're telling me a computer could figure out where the cells physically stood in three-dimensional space?
Yes. The math alone, based purely on those chemical attraction rules, accurately recreated the physical maps of healthy lymph nodes.
That is nuts.
It really is. It grouped the simulated B cells and T cells perfectly into their distinct zones, proving that the chemical signaling alone is sufficient to create the physical structure.
That is mind-blowing. The math proves that the chemicals literally are the architecture. The invisible web of signals dictates the physical shape.
And crucially, when they fed the computer the scrambled inflammatory chemical data from the DLBCL lymph nodes, the data where the street signs were gone and ectopic signals were firing.
Yeah, what happened?
The model perfectly predicted the fragmented, chaotic, diffuse architecture that we see in real patients.
Wow. For you listening, think about the magnitude of that. The physical shape of your immune system is completely dictated by an invisible web of chemical communication. When that communication breaks down, the physical structure literally dissolves.
It just falls apart.
But we always want to bring this back to the patient. Is this incredible mathematical proof just a fascinating biological quirk for textbooks? Or does this change things clinically for people actually fighting these diseases?
It changes our entire clinical perspective. The researchers took this chemokine data and analyzed a large cohort of real patient outcomes.
Okay, what did they find?
They discovered that this specific loss of homeostatic chemokines, this reprogramming of the city planners by chronic inflammation, it directly correlates with adverse patient survival.
So the patients whose lymph nodes lost their chemical street signs had worse survival outcomes.
Significantly worse. This identifies the architectural rearrangement not merely as a side effect of a tumor getting large, but as a key driving event in how the cancer progresses, how it evades the immune system, and ultimately how it impacts the patient's chance of survival.
Let's bring all these incredible insights together.
Yeah.
The core realization we're unpacking today is that an aggressive lymphoma, like DLBCL, it doesn't just lazily outgrow its environment.
No, it's very active.
It actively ensures its own survival by tricking the body's immune system into using its own emergency inflammatory signals to tear down the lymph node's structural integrity.
Understanding this structural collapse opens the door to entirely new ways of thinking about treatment. In biology, tissue structure is function.
Right.
The two concepts are inseparable. If you lose the physical structure of the lymph node, you lose the immunity. You lose the ability to coordinate the very attack needed to save the patient.
We spend so much time thinking of disease as a battlefield, but maybe the secret isn't just building better weapons to kill the invaders.
Exactly.
And for you listening, I want to leave you with a thought to mull over as you go about your day. Right now, so much of cancer treatment is focused on dropping bombs on the malignant cells, using chemotherapy or radiation to just kill the bad guys.
Which is very taxing on the body.
Super taxing. But if we now know that this aggressive cancer relies on a chronic inflammatory alarm to destroy the immune system's physical layout, well, could the future of treating these lymphomas involve city restoration drugs?
That is the big question.
Like therapies designed not to kill cancer cells directly, but simply to turn off the inflammation, letting the city planners sober up, put the street signs back up, and allow the lymph node to rebuild its own walls so it can fight the cancer naturally.
It's a completely new paradigm.
It really is. Think about that. We'll catch you next time on The Deep Dive.